Okay, everyone, welcome back to class. I just want to go through this walkthrough of this figure about how apoptosis works. This is what we watched in class. This is also available on the Pearson website if you have bought access to mastering, which is not a required component of this class, but I do think that mastering content is also always pretty nice to have access to, and I'd encourage you to get it if you're interested in it. Okay, here we go.
Hi, this is Jeff Harden from the University of Wisconsin in Madison, Wisconsin. Today I'm going to walk you through this figure showing two ways that apoptosis, or programmed cell death, can be induced in a target cell. While there are several pathways that can cause cell death, we'll be focusing on the two shown here, either driven by cell death signals or withdrawal of survival factors. The first pathway is an extrinsic one that begins when a cell death signal is received from outside the cell. These can be delivered in a number of ways, through a variety of different death receptors. Here we see a cell death ligand on the surface of a cytotoxic T lymphocyte engaging with the death receptor on a target cell. The binding of this ligand causes receptor subunits to aggregate and recruits pro-caspase 8. Caspases are the key family of enzymes responsible for activation of apoptosis. They are proteases that start out in the inactive pro form. Once activated through their own proteolytic processing, caspases cleave other targets. Caspases are classified as being either initiators or executioners, based on whether they start or finish the process. Thus, the inactive caspase 8 recruited to death receptors are considered initiator pro-caspases. The clustering of pro-caspase 8 at death receptors leads to its own cleavage and activation. This starts a caspase cascade, eventually causing the activation of executioner caspase 3. Active caspase 3 is directly responsible for the characteristic changes seen in dying cells. The other pathway is an intrinsic one, in which apoptosis is triggered indirectly from within. This pathway can start in many ways, including the absence of survival factors binding to cell receptors or through DNA damage. These pathways all stimulate the activation of death-promoting proteins that cause mitochondria to become leaky. Disruption of the integrity of the mitochondrial membrane can lead to the release of cytochrome C from the intermembrane space into the cytosol. The released cytochrome C can complex with the adaptor protein APAF1 and initiate a pro-caspase 9 to form a structure called the apoptosome. Once assembled, this complex activates caspase 9. One of caspase 9's targets is the executioner pro-caspase 3, which is then converted to its active form. As we saw with the extrinsic pathway, the intrinsic pathway also results in activation of caspase 3, which causes the activation of apoptosis. The targets of caspase 3 include nuclear lamins, cytoskeletal components, cell-cell adhesion proteins, and inhibitors of DNA endonucleases. Let's examine what happens next. Here we can see a cell in which apoptosis has been induced. As the executioner caspases are activated, its chromosomes condense and the cytoplasm is reduced in size. This cell will lose contact with neighboring cells, as well as the extracellular matrix while it shrinks. Eventually, its DNA is digested at regular intervals, the nucleus and cytoplasm become fragmented, and the cell extends numerous blebs. Ultimately, these remnants of the dead cell, called apoptotic bodies, are ingested by circulating phagocytic cells. As a result, this programmed cell death event is largely non-inflammatory. This helps avoid unnecessary damage to surrounding cells and tissues. The decision for the cell to undergo apoptosis is a highly regulated one. So far, we've seen how several pro-apoptotic proteins promote cell death. However, many anti-apoptotic proteins are involved as well. Here you see the anti-apoptotic protein BCL2 at the mitochondrial surface, blocking the release of cytochrome C. This inhibits the action of death-promoting proteins, preventing apoptosis. In other words, pro- and anti-apoptotic proteins, influenced by cell signals, wage an ongoing battle. Once the balance shifts, the cell's fate is likely sealed. Proper regulation of apoptosis is critical in all cells. Defects in these pathways can have profound impacts by altering embryonic development or promoting autoimmunity. Thanks for watching, and good luck with your course.
All right. Thanks for watching the videos. I will see you back in class. Bye.